For decades, people living with fibromyalgia, chronic fatigue syndrome, and Sjogren syndrome have been given some variation of the same routine response. We don't see anything on your test. It could be stress. It might be depression. Here's a referral. The wording shifts, but the underlying message stays constant. Your suffering has no biological foundation. Even people with Sjogren's, who frequently do have a positive blood test, hear a version of it too. Your labs explain the dryness, but the pain and exhaustion? That's likely something separate. We can't fully quantify it, so it must not be fully genuine. But here's what was actually unfolding. Science wasn't disproving these conditions. Science simply hadn't been searching in the right places, or studying enough people, until now. This video pushes 2 layers further, into the specific genes involved, into what NIH researchers actually detected when they examined the brains, spinal fluid, and immune cells of people with chronic fatigue syndrome, and into how Sjogren syndrome, an autoimmune condition with its own genetic signature, fits into this same biological territory of fatigue, pain, and immune dysregulation. Before we continue, I built something specifically not for just tracking and doctor's appointment. It is called Sjogren's Ultimate Symptom Map and Diagnostic Action Plan, and it walks you through exactly which symptoms to track not just for Sjogren but also for MS and RI. Drop symptom map in the comment, and I would send the link for you to access it. What the genetic studies uncovered. In August 2025, a team at the University of Edinburgh released findings from a project a decade in the making, the DECODE-ME study. They compared the DNA of 15,579 individuals with chronic fatigue syndrome against 259,909 people without it, over 250,000 participants, with contributions from more than 100 collaborators. It stands as the largest genetic investigation of this illness ever attempted, and its scale matters, because small studies simply don't have the statistical power to pick out faint genetic signals scattered across a condition this variable from person to person. Months afterward, a separate research group published the largest fibromyalgia genetic study to date, 2.5 million individuals, roughly 54,000 fibromyalgia cases. That's not a typo. 2.5 million people is an enormous sample by the standards of any genetic study, autoimmune or otherwise, and it gave researchers the statistical muscle to detect subtle genetic patterns that decades of smaller studies had simply missed. They identified 8 significant genomic regions tied to chronic fatigue syndrome and 24 linked to fibromyalgia. When researchers examined which tissues these genes were most active within, using tissue expression databases that map where in the body a given gene actually does its work, the answer was consistent across both studies: the brain. Not the muscles. Not the joints. Not some diffuse, unlocatable everywhere. The brain. That single finding appends a huge amount of outdated thinking about these illnesses. For years, the working assumption, spoken or unspoken, was that fibromyalgia and chronic fatigue syndrome were disorders of the muscles, or disorders of mood, or some combination of both, dressed up in physical language. What the tissue enrichment data says instead is that the genetic risk is concentrated in neural tissue, the organ responsible for interpreting pain, regulating arousal, and deciding how much energy to spend on any given task. That's an entirely different starting point for understanding these conditions, and it opens the door to an entirely different set of treatment targets. Sjogren syndrome carries its own, independently established genetic narrative, built across more than a decade of autoimmune genetics research rather than these 2 2025 studies. The strongest and most consistently replicated signal lies within the HLA region, HLA-DRV-DQ, the portion of the genome that governs how the immune system distinguishes self from foreign. This is the same broad genomic neighborhood implicated in other autoimmune diseases like type 1 diabetes and celiac disease, which makes sense. Sjogren's is fundamentally a disease of misdirected immune recognition, where the body's defense systems mistake its own moisture-producing glands for a threat. Beyond HLA, genes such as STAT4, IRF5, and BLK surface repeatedly across Sjogren's cohorts. All 3 sit within the interferon signaling pathway, the immune system's early warning mechanism for viral infection. In a healthy immune system, interferon signaling ramps up sharply during an infection and then settles back down once the threat has cleared. In Sjogren's, that alarm seems to remain locked in the on position, a phenomenon researchers often refer to as an interferon signature, elevated interferon-stimulated gene activity that's measurable in blood samples from Sjogren's patients even when they're not experiencing an active flare. So the CFS and fibromyalgia genetics point toward the brain circuitry for pain, arousal, and reward. The Sjogren's genetics point toward an immune system primed to attack the body's own moisture-producing glands, generating along the way the same kind of chronic low-grade inflammation reflected in the knee data discussed below. Different starting points. Overlapping downstream biology. And that overlap is exactly why so many people end up carrying more than one of these labels at once, wondering whether their diagnoses are coincidence or connection. What the genes reveal. Let's unpack what the CFS and fibromyalgia genes actually do, because the clusters tell a story, and it's worth slowing down on each one, because these aren't abstract data points. Each gene corresponds to something you can likely recognize in your own daily experience. CA10. The volume knob. The clearest signal across the entire DECODE-ME study, a single gene, one chromosome, no competing candidates, was CA10. It plays a central role in brain development and central nervous system function, and it's already been independently tied to widespread chronic pain and insomnia in research completely separate from this study. When a signal shows up this cleanly, with essentially no competition from other candidate genes on the same chromosome, researchers pay close attention, because it suggests a strong, specific biological effect rather than statistical noise. UNC13C and SNAP25, the glutamate problem. UNC13C and SNAP25, loosely referenced as SSA6 in earlier drafts of this script, are both involved in glutamatergic synaptic transmission, the process by which neurons release glutamate, the brain's primary excitatory signal, across the junctions between them. Think of every neuron in your brain as constantly deciding whether to speak up or stay quiet. Glutamate is the chemical equivalent of speaking up. It's what allows one neuron to excite the next, building the cascades of activity that let you notice a touch, register a sound, or feel a stab of pain. When the genes governing glutamate carry variants, the nervous system loses the ability to regulate its own arousal. It remains stuck in excitatory overdrive, pain that never switches off, sensory input amplified well beyond what the stimulus warrants. Picture it as the volume dial for your body's entire pain and threat response system cranked all the way up, with no hand available to turn it back down. A light touch registers as pressure. A dim light feels harsh. A quiet room still hums with noise your brain refuses to let go of. COMT, the dopamine drain. The fibromyalgia study contributed something that reframes the motivational struggle entirely. Its strongest tissue associations centered on the basal ganglia, the caudate, the putamen, the striatum. These regions anchor the brain's dopamine circuitry, and they are nodes within the broader network governing attention and reward. A gene called COMT produces an enzyme that breaks down dopamine in these regions, particularly within the prefrontal cortex. One specific variant, VAL158MET, appears across both fibromyalgia and chronic fatigue syndrome populations in multiple independent studies, a shared genetic vulnerability where cognitive fatigue, motivational paralysis, and sensory amplification converge. People carrying this variant tend to break down dopamine faster in key brain regions, meaning the reward and motivation signal that should accompany completing a task, or even just anticipating something enjoyable, gets metabolized away before it can do its job. This is why the motivational flatness, the inability to experience reward, the exhaustion that doesn't respond to antidepressants, isn't a character flaw. It's embedded in the dopamine architecture of these brains, and no amount of willpower changes the chemistry. OLFM4, the insomnia thread. A third gene, OLFM4, contributes to neural development and synaptic function, and it's independently linked to depression and insomnia. CA10, SNAP25, and OLFM4 sit on 3 distinct chromosomes and are each independently tied to insomnia, not sleep disruption as a side effect of pain, but insomnia woven into the genetic architecture from the outset. This distinction matters clinically. If insomnia were purely downstream of pain, then treating the pain should fix the sleep. But if insomnia has its own independent genetic roots, that explains why so many people with these conditions report that even on their best pain days, on days when nothing hurts especially badly, sleep still doesn't come easily, and when it does come, it doesn't restore them the way it should. RAPGAP1L, the infection connection. There's also an immune and viral signal, RAPGAP1L, active across a broader range of tissues than nearly any other gene identified in the study. It helps suppress viral replication inside cells, a direct genetic pathway toward understanding why chronic fatigue syndrome so frequently follows an infection. Anyone who has ever had a mild viral illness spiral into months or years of unrelenting fatigue has likely wondered whether it was really just a virus, or whether something changed permanently in the aftermath. This gene offers a partial, biologically grounded answer. Some people appear to carry a genetic predisposition that makes the post-infection recovery process go wrong at a cellular level. Researchers also flagged variants near an immune checkpoint gene, though the precise mechanism there remains under investigation, an area likely to see rapid progress in the coming years as more samples become available. That final thread is where Sjogren's rejoins the narrative. Sjogren's is, fundamentally, a disease of immune checkpoints and viral response machinery running into overdrive. The interferon pathway genes, STAT4, IRF5, sit adjacent to that same kind of immune system that won't stand down. Biology researchers are now finding hints of within RABGAP1L and neighboring checkpoint regions. Different genes, same neighborhood of dysfunction, an immune system that keeps battling long after the original threat has passed, whether that battle shows up as unrelenting fatigue, as dry eyes and dry mouth, or as both at once. What NIH scientists discovered inside living bodies. Now for the study that, in my view, ranks among the most important ever published on this community, and it didn't emerge from a DNA database. It came from researchers putting 17 people with chronic fatigue syndrome through the most thorough biological evaluation ever conducted on this illness. 17 people sounds small compared to the millions in the genetic studies, but this was a different kind of research entirely, not a search for statistical patterns across huge populations, but an intensive, deep-dive physiological workup of each individual participant, the kind of study that's extraordinarily expensive and logistically difficult to run at scale. Published in February 2024 in Nature Communications, led by Dr. Ravindra Nath, Clinical Director at NINDS, with 75 researchers spanning 15 knee institutes and over 8 years of effort, brain scans, spinal fluid analysis, immune cell profiling, muscle biopsies, 24-hour heart rate monitoring, metabolic chambers, cardiopulmonary exercise testing. Every biological system measured simultaneously in the same individuals, compared against matched healthy controls. Participants were admitted as inpatients for extended stays, allowing researchers to track changes over time rather than capturing a single snapshot. This is, by any reasonable measure, one of the most exhaustive biological investigations ever conducted into an illness that for decades was treated as if it had no biology worth investigating at all. The brain, and what effort truly represents. Knee researchers used functional MRI to examine how the brains of people with chronic fatigue syndrome make decisions surrounding physical effort. They discovered reduced activity in a specific region, the right temporoparietal junction, RTPJ. The RTPJ performs two functions, and both were compromised. First, it's part of the brain's bottom-up attention system, the automatic switchboard that scans for anything unexpected and redirects attention without deliberate effort. This is the system that lets you notice, without trying, that someone just called your name across a crowded room, or that the smell of smoke means something needs your attention right now. Second, it functions as a sensory gatekeeper. It integrates internal bodily signals and external sensory input and determines what passes through versus what gets filtered out. When functioning properly, it lets you sit in a cafe, surrounded by conversation, clattering cups, and background music, and still hold a conversation with the person across the table without being overwhelmed. When underactive, as measured here, the gatekeeper falls silent. Every background hum, every clothing tag, every minor ache gets processed rather than screened out, because the system responsible for deciding this doesn't need your attention simply isn't doing its job. In ADHD, researchers have repeatedly documented reduced cortical thickness and atypical functional connectivity within this same region, not a coincidence, but a shared anatomical vulnerability. Lay a chronic fatigue syndrome onto an already compromised RTPJ, and the underactive gatekeeper fails to properly drive the motor cortex during exertion tasks. The outcome, in the researcher's own words, is a shift in effort preference, not diminished effort. Grip strength remained intact. Muscles showed no evidence of fatigue on direct physiological testing. The dysfunction originates upstream, within the circuit that predicts what effort will cost before the command to move is ever issued. In plain terms, the muscles can do the work. The brain has decided, based on a miscalibrated internal forecast, that the work isn't worth what it will cost. As lead author Dr. Brian Wallet described it, rather than physical exhaustion or lack of motivation, fatigue may stem from a mismatch between what someone believes they can accomplish and what their body can actually deliver. That reframing matters enormously for how this illness gets talked about, both by doctors and by the people living with it. "You're just deconditioned, you need to push through it," assumes the problem is muscular. "You're afraid of activity," assumes the problem is psychological. Neither of those addresses a brain circuit that's making an inaccurate prediction about cost before any decision to move has even been consciously made. For Sjogren's patients, this carries direct relevance. Fatigue is consistently identified as one of the most disabling symptoms of the condition, often ranked by patients as more burdensome than the dryness itself, and multiple studies have found that Sjogren's-related fatigue correlates only loosely with disease activity markers like inflammation or gland damage. In other words, a Sjogren's patient can have well-controlled blood work, minimal visible gland inflammation, and still describe fatigue severe enough to derail their entire day. That disconnect mirrors exactly the pattern the RTPJ findings help clarify. Fatigue that doesn't track tissue damage may instead track a brain circuit that's miscalculating the cost of effort, regardless of what's driving the underlying inflammation in the first place. The spinal fluid, what's depleted in the brain's chemistry. NIH researchers measured catecholamines, norepinephrine, dopamine, serotonin, and related signaling molecules, directly within cerebrospinal fluid, the fluid that bathes the brain and spinal cord. This is a meaningfully different kind of measurement than a standard blood test. Blood levels of these chemicals don't necessarily reflect what's happening inside the central nervous system, because the blood-brain barrier restricts what moves freely between the bloodstream and the brain. Sampling cerebrospinal fluid directly gives a much more accurate picture of the chemical environment the brain is actually operating in. Levels were abnormally low relative to healthy controls, and the reductions correlated with worse motor performance, worse effort-related behavior, and worse cognitive symptoms, meaning this wasn't a stray finding sitting off to the side, but a measurement that tracked cleanly with the severity of what participants were experiencing day to day. Dr. Nath framed it this way: "It's like having the flu, where your body's nervous system tells you to lie down and not exert yourself. Except for people with chronic fatigue syndrome, that signal never switches off." Anyone who has had a bad flu knows that particular, heavy, whole-body instruction to stop moving. Now imagine that instruction arriving every single day, regardless of how much rest you've had, because the chemical systems responsible for eventually switching it back off are running short. The autonomic nervous system, a body that never fully recovers overnight. Researchers tracked 24-hour heart rate continuously across all participants. People with chronic fatigue syndrome exhibited significantly reduced heart rate variability, a well-validated marker of how flexibly the nervous system can shift between go and rest states, along with elevated heart rates throughout the day, and a blunted nighttime dip in heart rate. That nighttime dip is supposed to happen when the parasympathetic system, the rest and digest branch of the nervous system, takes over during sleep and allows the body to genuinely recover. In these participants, it barely happened at all. The formal tilt table test, which checks for a sudden drop in blood pressure or spike in heart rate upon standing, revealed no group difference in orthostatic hypertension or excessive orthostatic tachycardia. That's an important negative finding, because it tells us this isn't simply a case of a single, dramatic cardiovascular event. Instead, what surfaced was something quieter and more pervasive, a chronic, low-grade imbalance in autonomic tone, persisting around the clock, day and night, whether the person is standing, sitting, or lying in bed trying to sleep. This is one of the clearest overlap points with Sjogren's. Autonomic dysfunction, small fiber neuropathy, abnormal heart rate variability, orthostatic symptoms, has been repeatedly documented in Sjogren's patients, independent of standard disease activity scores, and is believed to significantly contribute to the fatigue and diffuse pain many Sjogren's patients report. Small fiber neuropathy in particular is increasingly recognized as a genuine, measurable driver of widespread pain in Sjogren's that doesn't register on a standard nerve conduction study, which typically only picks up damage to larger nerve fibers. That's part of why so many Sjogren's patients describe burning, tingling, or diffuse aching pain that gets waved away because the nerve tests came back normal. The tests being used simply weren't built to detect the kind of damage involved. The immune system, still battling something. Immune profiling within the CFS cohort revealed elevated naive B cells and depleted switched memory B cells, a signature of an immune system stuck in an early, nonspecific inflammatory mode, with T-cell exhaustion markers present. To translate that, naive B cells are the immune system's generalists, ready to respond to almost anything but not specialized for any particular threat. Switched memory B cells are the specialists, built from prior exposure to specific pathogens, capable of mounting a precise, efficient response. An immune profile skewed toward naive cells and away from memory cells looks like a system perpetually caught in its first, clumsy response to a threat, never quite graduating to the more refined, targeted defense it should eventually build. T-cell exhaustion, meanwhile, is a state where immune cells that have been fighting for too long start to lose their functional capacity. They are still present, still technically on duty, but increasingly unable to mount an effective response. It's a pattern seen in chronic infections and some cancers, where the immune system has been fighting the same battle for so long that its cells begin to wear down. Researchers interpreted this as chronic antigenic stimulation. The original trigger may have long since cleared, yet the immune system continues responding to something, generating low-grade neuroinflammation that feeds back into the brain circuits governing effort, dopamine, and autonomic tone. Sjogren's is, by definition, an immune system that won't stand down. Lymphocytes infiltrate the salivary and lacrimal glands and, in many patients, other organs as well, including the lungs, kidneys, and peripheral nerves. The interferon pathway genes, STAT4, IRF5, that appear in Sjogren's genetics align closely with exactly this kind of chronic, low-grade immune activation. It's a different underlying mechanism than what's proposed for CFS, an established autoimmune process rather than a hypothesized post-infectious one, but the downstream effect, inflammation that continues signaling to the brain long after any acute threat has passed, looks remarkably similar. Fibromyalgia, Sjogren's, and the connecting genes. Two genes surface in both the chronic fatigue syndrome and fibromyalgia studies, OLFM4 and DCC. DCC guides the axons of dopaminergic neurons, the same reward and motivation circuits the NIH study found depleted, toward their destinations during brain development, and it's independently linked to pain, depression, and autism spectrum conditions. A gene involved in wiring the dopamine system during fetal and early childhood brain development showing up independently in both illnesses suggests that some of this vulnerability may be laid down years, even decades, before the first symptom ever appears. Sjogren's doesn't share these particular genes within the published data. Its genetic story runs instead through HLA and interferon pathway genes. But clinically, the overlap is substantial and well documented. Studies estimate that a considerable proportion of people with Sjogren's also meet criteria for fibromyalgia, and fatigue is reported by the vast majority of Sjogren's patients regardless of how controlled their condition appears on paper. This is precisely the kind of comorbidity pattern that gets dismissed in a 10-minute appointment as, well, autoimmune disease is exhausting, that's expected, without ever asking whether there's a distinct, parallel condition layered on top of the primary diagnosis. A 2024 study from Yale analyzing 6 chronic overlapping pain conditions, fibromyalgia, IBS, migraine, endometriosis, TMJ disorders, and chronic low back pain, found evidence of a shared polygenic vulnerability spanning all of them, not 6 distinct diseases, but one nervous system vulnerability manifesting across different bodily regions, depending on where in the body that underlying sensitivity happens to express itself. Sjogren's wasn't included in that specific analysis, but its symptom profile, widespread pain, unrefreshing fatigue, small fiber neuropathy, autonomic dysfunction, fits squarely within that same cluster, and it raises a genuinely open scientific question worth watching. Does an autoimmune trigger like Sjogren's activate a pre-existing nervous system vulnerability, the same one implicated in fibromyalgia and chronic overlapping pain conditions? That question doesn't have a settled answer yet, but it's exactly the kind of question this new generation of large-scale genetic and deep phenotyping research is finally equipped to test. What this means for you. So what does this actually mean, sitting at home with a diagnosis, or 2, or 3, that still leaves most doctors puzzled? First, you are not imagining this. The NIH measured it directly, a specific dysfunctional brain region, depleted spinal fluid neurotransmitters, an autonomic nervous system unable to rest, immune cells locked in a chronic struggle. This isn't a theory pieced together from anecdotes. It's measured human biology, published in one of the most respected scientific journals in the world, produced by one of the most rigorous research institutions on the planet. And if you have Sjogren's, your blood work already confirmed your immune system is doing something real. The fatigue and pain accompanying it aren't automatically something else, some separate, unrelated mystery laid coincidentally on top of a disease that's already been proven. Second, these are treatment targets, not vague suggestions. Researchers pointed toward immune checkpoint interventions, approaches to support depleted catecholamines and RTPJ function, and dopamine-supporting strategies similar to those used in ADHD. None of this means a cure is sitting on a shelf waiting to be picked up. These are research directions, not finished treatments, but it does mean the path forward has actual biological targets rather than a shrug and a referral to a generic wellness program. For Sjogren specifically, addressing interferon pathway inflammation and the documented autonomic and small fiber involvement, not merely gland dryness, represents an active and expanding area of research, with several interferon-targeting therapies already being studied in autoimmune disease more broadly. Third, the broader picture emerging across chronic pain, fatigue, and autoimmune conditions is one of overlapping, sometimes shared, sometimes parallel biological vulnerabilities, not a collection of separate, unrelated diagnoses, and never something willpower could have overridden. If you've spent years wondering whether your fibromyalgia, your chronic fatigue, and your Sjogren's are three coincidental things that happen to be happening to the same unlucky person, the emerging science suggests something more coherent. Overlapping vulnerabilities in the same handful of biological systems, the brain's pain and reward circuitry, the autonomic nervous system, and an immune system that has trouble standing down, expressing themselves through more than one diagnostic label at once. A note on what this isn't. None of this research suggests that these conditions are untreatable, permanent life sentences with no room for improvement, nor does it suggest that lifestyle, pacing, medical care, or support don't matter. Measured biology and manageable illness aren't opposites. Understanding the mechanism is the first step toward eventually intervening on it. That's true of essentially every disease in the history of medicine, from diabetes to cancer to heart disease. The point of this research isn't fatalism. It's legitimacy, and a map of where future treatments are most likely to come from. Your pain is genuine. Your fatigue is genuine. Your dryness and your exhaustion aren't two unrelated problems. And now we have genetic architecture, brain scans, spinal fluid measurements, and immune cell data to help explain why. If this video has helped you, pass it along, to someone who's been dismissed, to a family member who doesn't quite believe you, to your doctor. Every person who encounters this science becomes a little harder to dismiss. A word on talking to your doctor about this. Bringing a stack of printed studies into a 10-minute appointment rarely goes well, not because your doctor doesn't care, but because most clinicians simply haven't had time to read primary literature on a condition that, historically, medical training spent very little time covering. A more effective approach is often to ask specific, narrow questions rather than presenting the whole picture at once. Has autonomic testing, like heart rate variability monitoring, ever been considered for me? Given the overlap between Sjogren's and small fiber neuropathy, is that something worth screening for? I've read that fatigue in these conditions doesn't always track with standard inflammation markers. Does that change how you'd interpret my labs? Questions like these invite a conversation rather than a confrontation, and they give a doctor who is willing to engage something concrete to respond to, rather than asking them to become an overnight expert in a still emerging body of genetics and neuroimaging research. It's also worth remembering that this science is moving quickly, and it's moving in your favor. 5 years ago, a study of this depth on chronic fatigue syndrome, with this many institutes, this many researchers, and this many overlapping physiological measurements taken from the same small group of patients, would have been difficult to imagine funded at all. The fact that it exists, and that it was published somewhere as prominent as Nature Communications, is itself a signal that the scientific and medical establishment is beginning to take these conditions seriously as measurable, biological phenomena rather than as diagnoses of exclusion handed out once every other explanation has been ruled out. Share your story in the comments. What's the one thing you wish your doctor understood about the biological basis of your illness? Let's turn this comment section into a genuine resource. Thanks for watching.